going to represent my presentation on genetic markers. Here's a table of content. What is marker, type of marker, genetic marker, application of genetic marker, and reference. What is marker? Marker are used for assessment, identification of diversity and polymorphism result diversity. For example, the multiple color of fruits, flowers, and seed in species. This diagram shows that the marker have three types. Morphological marker based on diversity for our traits, not need lab study, affected by environment, require a proper stage and time to study. Second one are molecular markers. They are also called DNA markers and genetic markers. These DNA markers are further divided into non-PCR base and PCR base. Non-PCR base RFLP, restriction fragment length polymorphism. It was first molecular marker. In RFLP, the first isolate the DNA, then mix with the restriction enzyme and run on gel. The PCR based markers, first rapid, the random amplified polymorphic DNA in rapid oligonucleotides are used. The single stranded DNA sequencing used in PCR technique. Primer is used to hybridize with DNA sample. Actually, the molecular marker are based on diversity in DNA sequencing, and the AFLP is a amplified fragment length polymorphism. In AFLP, markers combine with RFLP and PCR technique. Biochemical markers, non-genetic markers, first isolate the protein and run on the gel. The genetic markers, molecular markers are the nucleotide sequencing and can be investigated through the polymorphism present between the nucleotide sequencing of different individuals. An ideal DNA marker should be codominant, evenly distributed throughout the genome. The marker assists selection for desirable trait and genetic mapping and quantitative traits analysis. The genetic and genomic association study and gene expression analysis and forensic sciences, DNA profiling and identification. Here is the reference and the last one slide for thanks.